So I'm going to talk about just detecting structure in neuronal networks, although this title is a bit of a lie because towards the end, if we make it that far, I will talk about some non-neuronal brain networks. So I should have used brain here, but whatever. Okay, so, and of course, before I get started, I need to acknowledge that none of my work is my own. Vladimir and Karina and Eva Postelkova and I worked on the primary thing that I'm going to be discussing throughout this talk. And then more recently, I have a variety of collaborations with people at Penn who are doing fantastic work, and that'll come later if we get there. However, I also want to say that I really do want everyone to understand every single thing that I say in this talk. Everyone in this room is totally, there's nothing hard. So we don't have to get to the end of the slides, okay? So ask questions. Stop me if there's anything you want to ask about. And then also, algebraic topology in neuroscience has an incredibly small intersection right now. So I have a web page that contains all of the papers that I'm aware of. And I would encourage you to take a look if you find this stuff interesting. It is a little out of date. I haven't updated it in a couple of months, and in particular, one of my papers isn't on there. But I'll put a link to that at the end. So I'm going to use another Lichtman Lab picture because these things are so fantastic. And I'm going to make a really controversial statement to get your attention. Brain networks are complicated, and they're hard to understand. And just looking at this picture, you say, well, I guess we should probably just go home. And I tend to agree with that. So this is another one of those smaller than a grain of sand chunks of mouse cortex. It contains hundreds of different cells. It was a massive engineering effort just to get this picture to exist. Right? They had to slice this thing and then take into these incredibly tiny pieces and then take electron microscopes and scan each one of them and then use software that they, painstakingly built for years to reassemble those into cells. And you say, well, okay, maybe there's hope. But then you look at the pieces. So here we took out the two major big arterial pieces there. Those are just the surrounding bits. And it just, okay, so we're not going to start there. This is something that hopefully you all can solve when you get involved with these problems, which you all want to at the end. So I'm going to focus on function first, because that's something I have some hope of understanding. And the reason I have some hope of understanding it is because I have a cheat sheet. So this guy, John O'Keefe, who you may have heard of, won the Nobel Prize last year in medicine, joint with the Mosers, for his discovery of place cells in the hippocampus in the 1970s. So the idea is that you have a mouse or a rat, and it's moving around its environment, and you put a probe in its hippocampus, and you record, so this black is the trail, and every time a particular neuron in there spiked, put a red dot at the point where the rat was sitting, or standing, right? And what do we see? Well, this one neuron in the hippocampus is coding for location, right? This one spot in space. That's fantastic. But the hippocampus is nowhere near your sensory systems. It's like one of the deepest brain areas that actually has neuron bodies in it. The sensory information is completely destroyed by the time you get there, right? It's been disseminated, processed, but somehow there's this one cell deep in your brain that's literally coding for position. And if the rat moves over here, it'll be a different one. If the rat moves over here, it'll be a different one. So that's, wow. That's great. That's a cheat sheet, right? Now I know what that area of the brain is doing when the rat's navigating. There are cells in there, and they're recording location. Okay. So nobody believed O'Keefe when he first said this happened. It was a long uphill battle to get people to believe this was really occurring. They thought it was noise, that it was, right? Because he could also only record from a single unit. He had very, very bad technology compared to today when we can get hundreds of simultaneous units and build a complete map, right? 73, I think, was the first paper. 74. So, yes. Yeah. because you can do it. I've seen it in the lab. I mean, so you can get, so here, I'll show you some pictures. So these are from an experiment that Eva Postolkova did with rats, and they were we've been replicated tens of thousands of times, and it's not your grandmother, right? It doesn't matter what environment you're in, there are cells corresponding to location, which makes some sense, because unlike evolving a cell that responds specifically to your grandmother, it makes extreme sense for your brain to be able to represent where it's at, right? So this is a much more, also, yeah, we've 40 years now, and it's just gotten more and more and more complete, our understanding of how these things work. Right, so these are 3 cells, actual data. What we did is we just convolved, right, spiked with location, like a little Gaussian, right, and then we normalized for the amount of time the rat spent in that location. We got these nice heat maps that tell you, approximately where the cell is responding. And so they're not perfect, but they're pretty darn good. These things are called place fields. Okay. So how do I think of this as a network? Because I told you I was going to think about function and networks. Well, there's a standard thing neuroscientists like to do, and a lot of people like to do, right, which is to take activity of, right, so what are these? These are cell responses, right? This is my conceptual map up here of what the cell is doing that John O'Keefe gave me. Here's what I can actually observe, right? What do I observe? The neurons are firing, right? So there's getting activity. And 2 different neurons have different activity, and so I can compute a cross-correlogram for those 2 time series, right? And then I can take, let's see. So I've got, if they were just randomly firing, I just have the cross-correlogram be the product of their firing rates, so that's sort of my baseline. And here I see that these 2 things are, right, the mean of that cross-correlogram is higher than the baseline. That suggests to me that these 2 things are in fact correlated. Okay, so I'm just going to record that value, that Cij, in a giant matrix, and this is the data I'm going to pretend that you've given me, right? Is you've handed me a matrix. Oh, a matrix, a weighted matrix. I will conflate that with a network, because that's what I do, because I'm a mathematician and I can't see the difference between things. So that's a correlation network, right? So this is my starting point. I'm going to imagine for a moment that I live in a world where John O'Keefe never existed, and so no one has given me my cheat sheet. Except I can't really quite get there, so I'm going to use the cheat sheet all along the way as intuition. But what I'm really thinking towards is that there are actually about 3 brain regions that we understand even the slightest amount of. We know a little bit about the hippocampus. We know there's associative memory because of an unfortunate neuroscience or neurosurgery experiment. And we know that it has to do with location because of O'Keefe. We know about vision because we're really good at vision, right? We can show you things. And we know a little bit about motor cortex, but not very much. There are arguments about the basic coding properties of motor cortex. So that corresponds to like here and here and one little chunk right in there. But if I choose about 75% of the surface of your brain and stick a probe in it, I don't know what I'm supposed to be seeing, right? So that's really the thing, right? You're likely to give me a matrix, and I don't know what the function of the network is supposed to be. So my question is, how in the world, given that matrix, could I see that maybe this was happening? Okay. So here's a conceptual model, right? So I just want to break down the conceptual model a little bit, right? So this first neuron is centered here. Second neuron has a place field centered here, and the third place field is centered there, right? And I've got this correlation matrix. So which of these things are going to be more highly correlated? Well, we have a conceptual model, right? So, right, the distance between 1 and 3 is smaller than the distance between 2 and 3, which is smaller than the distance between 1 and 2. So the correlation should roughly line up like that, right? Assuming that I've sampled the system well. So when I'm going forward, actually, this is going to be the thing I hang my hat on. Okay. However, there is a serious confound here, because you might say, let me take my network, right? I know I'm looking for geometry. It's a network, so that should be giving me distances. I'm going to go get MDS. I'm going to go get isomap. Why are you bothering with all this hard work, right? I'm going to just flatten it down into low dimension and see if it fits. Neural responses to stimuli are nonlinear. So the farther away you get from that point, right, the response doesn't drop off from that center point in a linear fashion. And therefore the correlations between the activities are not going to be a linear function of distance. It's going to be some bad nonlinear function of distance. And although it's going to be a bad nonlinear function of distance, I'm never going to know what that function is. I can't possibly know, because it's different for, this is very different for different systems, very different for different neurons. So I'm in trouble. And this is going to be nice. It's going to be our saving grace. Measurable quantities increase monotonically with the thing I actually care about. With actual distance, the correlation should go down. Right? So the quip is that biology is nonlinear, but it's not malicious. Mostly. So if I wanted to go to my standard network toolbox and pull out my PCA or my favorite other thing that I would use to detect the existence of structure, right? So I like to pick on Wigner's semicircle law because it's such a pretty theorem. So here's a 1,000 by 1,000 random matrix with IID Gaussian entries. And if I compute the eigenvalues, right, this fundamental theorem in random matrix theory tells me that the eigenvalues lie in this range that I can compute, and their distribution is this beautiful semicircle. What do you see? A semicircle distribution, right? So I see that and I go, oh, matrix is random. Cool. But what if I take that same matrix and I pass it through a saturating nonlinearity? And then I didn't put it on the equation, but I'm also going to zero mean it, so I'm back in Wigner's semicircle land. Okay? So this is not an unreasonable thing, right? This is just an exponential fall off. So I took that same matrix, I passed it through there, I go over here and I compute the eigenvalues, and whoa, principal components. This is a rank 5 matrix. Cool. There's structure. But Chad, you say it's an IID matrix. You just chose from different distribution. What happened? Well, these long-tailed distributions, these saturating nonlinearities, do terrible damage to the convergence properties of these things. Even in a 1,000 by 1,000 matrix with this simple nonlinearity, I'm able to make it so that you haven't reached the convergence range for Wigner. And of course, when are you going to see more than 1,000 cells in the lab? Well, the Obama Brain Initiative's working on 1 million, but it's going to be a while. So, well, no, it's not. It's IID, right? It's not rank 5, but it sure looks rank 5 if you just glance at the eigenvalues, right? Right? Like if I'm a first-year grad student, wander into the lab, and I'm like, haha, rank 5, I'm going to write a paper. This is great. I found structure.
I see
that long junk in the middle. Yeah, but that could just be noise around 0. Right, I'm just saying these things look like they're outstanding to me, right? If I plot this thing and I look for what's the serious structure, I see 5 things, right? That stuff in the middle is complicated, but the stuff on the outside is not. Those are principal components or something. So this is dangerous. Nonlinearities damage eigenstuff, and they should. Eigenstuff is what's invariant in the matrix under linear transformations, under change of basis. Nonlinear stuff, all bets are off. So how can I possibly detect structure under these terrible, terrible conditions that I've imposed by being a biologist? Well, I claim algebraic topology is the key. And that is not only because I have a background in algebraic topology, but it is partially because of that. I think it's just amazing. If you were a biologist and you sent a wish list back in time to Poincaré and said, invent a field that does this. So what did he invent? A field that is robust to noise, right? Topology is all about continuous deformation. I wiggle systems, they don't change. Great, cool. So I'm robust to noise, that's great. And I can force tools to ignore other things, like say your favorite nonlinearities, and that's what we'll do in a moment. I can study objects through the lens of how they're related to each other instead of how they exist in a vacuum. So this is, a lot of people here have talked about graphs, and graphs are our fundamental starting point, right? I'm talking about networks. They're a tremendous tool. There's one real problem with graphs in my mind, and it is this. If I give you 2 graphs that are supposedly related, same system, different times, maybe even a map between the 2 graphs, right? This graph has a graph morphism to that graph. And then you compute your favorite graph invariant on graph 1 and your favorite graph invariant on graph 2. And they're different. Can you tell me why they're different? Algebraic topology is designed as a field, so that if I give you two objects and a map between them, whatever I computed over here, I can carry over to there and check. That's what it's for. So, right, and everything in biology is about interrelationships. Everything in neural networks is certainly, right, in the bigger field of neural networks. It's all about relationships. So, cool. And then lastly, the data you get back in topology tends to be qualitative, right? The standard question topologists ask are how many holes are in this object, which is not, you get a quantitative answer, but what's a hole? And it turns out that answer is really hard. So, in fact, I'm not even going to try to tell you what the answer is during this talk, but we had to work really hard to make that definition. But then if you show somebody a picture of a hole, they go, yeah, that's a hole, right? So it's a qualitative thing. And we build that out of local information. The hole is a global thing, right? At no point when I'm building a circle, before I get to the end of the circle, do I have a hole in it. I have to have the whole thing. Okay, so that's a global structure built out of local objects. And again, biology, all about that stuff. So I claim this is the right set of tools. At least one right set of tools. Of course, people have done tremendous things with other tools. New tool. Okay, so I'm going to tell you about three things from algebraic topology. Most of these things are actually from algebra, but they were applied by topologists in this particular way. And they're all really simple. So filtrations are a way to take a weighted object and give you an unweighted object without throwing away the information. Standard thing to do in biology is threshold, right? These things are important, these things are not important. I don't like to do that because I don't know how to make decisions because I'm just a mathematician, and it often feels like maybe I'm throwing away the wrong stuff. For example, one of Daniel Bassett, one of my postdoc mentors' recent papers shows that the 20% of the weakest connections in the human brain tend to have a stronger cognitive effect in terms of predictive value, because the super strong stuff is stereotyped, and the weak stuff is what varies from person to person. But if you thresholded, that would not be the thing you would guess, right? So, so you're likely to throw things away. So if we have a principled way not to do that, that's cool. Filtrations are our way to do that. Simplicial complexes are just a way to take networks and soup them up a little bit. And essentially we're going, right, networks talk about dyads, right? This thing is related to that thing. But not these 17 things are all related. And I have no way in a network to distinguish between whether all the pairs are related individually or whether it's one giant group relation. Simplicial complexes solve that problem. And lastly, persistent homology is a way to study the structure of simplicial complexes. It's where those holes are hiding. None of these things are complicated ideas. And I'm going to show you how to get going. But I want to tell you why I think this is the right tool to solve that first problem, which is I'm watching the brain, and I think maybe there's geometry hiding in there. So let's imagine I have a giant network, but I'm only looking at these. For now, I'm just going to look at these 4 neurons. Okay? So I've arranged them so that their distances are supposed to be the actual correlation, because when we draw a graph, we intrinsically, we've accidentally added geometry to it, so I may as well exploit that. Okay? So let's see. If I'm adding in terms of strength of correlation, well, first the vertical nodes are going to be connected, so I'll just stick edges there to tell you those things are close together by my current notion of close together. Let's see. Okay, now, of course, I'm going to get the horizontal lines. Okay, what have I done? I've created a square. That's a hole, in case that thing I didn't want to define for you, that's a hole. Okay, question. How long has that hole got to live if I continue to increase my parameter? Not long, right? It's got to come along pretty soon. Why? Why does the cross edge have to appear? High school geometry. Right. This edge squared plus this edge squared. It's the triangle inequality. It's one of the 3 things you assume when you say you have a metric, right? is the triangle inequality. So this big square cannot persist long once it exists in the presence of a metric. But if there is no metric, I have no way to predict how long it's going to exist, right? If there was no metric structure going on, I could have this structure show up and then just sit around forever because that length in the middle might be 1 billion times as long as everything else. Okay? And that's what I'm going to shoot for, is detecting that kind of difference. Right? It turns out persistent homology is just like custom made to do this. Okay. Step 1, filtrations. Filtrations are so simple that I actually feel bad showing you slides. Here's my favorite 15-node network, and I've used JET because that was the color scale that I chose. You can all agree, I think we can all agree that JET is a terrible choice. If you've not seen the Python scikit-learn people's talk about color choices and why JET kills, you should. It's actually, I mean, they make it as a joke, but then they go on and say, look, this actually happens. Doctors make misdiagnoses because of JET. So don't use JET. All right, I have a strongest edge. Maybe that's the thing I should include, right? Maybe I should threshold there. Okay, so there. Now I've included the strongest edge. Was that the right choice? I don't know. So let me include the second strongest edge. That seems like a good threshold. Well, did I make the right choice? I don't know, right? Like I said, I don't know what to do. I don't know how to make choices. So what will I do? I will make all the choices. Right? Simple enough. So I'm just going to take all the possible threshold, all the possible binary graphs I can get by thresholding this graph. Okay? What have I got? I've got a sequence of graphs indexed by decreasing edge weight, right? They're all sitting above on this axis above the weight of the edge. So I've still got all the edge weight information. But each one of these is a binary graph, and I can use all my favorite binary graph tools on it. Right? And we all love that kind of stuff because it's easy to compute. We've got a giant toolbox. Further, each one of these includes into the next one. Right? I've just added one edge. So all I've done is I've taken your giant ugly graph up there, and I've unfolded it into a sequence of graphs indexed by the weight at which I thresholded. And that sequence of graphs contains a canonical choice of inclusions. Good? See, I told you this stuff is like there's nothing to it. What's that? No, I don't break ties. If two numbers are the same, then I put in the edges at the same time. However, that will never happen in a data matrix. So for the mathematicians in the room, I don't break ties. For the biologists in the room, why would I ask that question? I never would. So, all right. So, okay. So now I can compute my favorite graph invariant, whatever you like, my favorite measurement of graph structure for each one of these things. And whoa, what do I get? I get a function. Okay? A piecewise linear function or a stepwise function or whatever you want to make it. Okay? So I've taken, I don't know, let's say, number of components. All right? And now I can compute it for every graph. And now, instead of getting some individual picture of a threshold with the number of components, I've got this sort of concave looking thing. Okay. This is what I like to refer to as a second order graph invariant because it works on weighted graphs and it tells me something more than any given threshold. In fact, if I took a collection of 3 or 4 thresholds, I might still not get the whole picture I've got here because this concavity of this function actually turns out to tell me something that, as far as I can tell, is a hallmark of modular graphs. So where I've got strong modules connected by weak edges. I'm not going to go into that at all. That's Chad ran some simulations because he wanted to make some pictures. I don't have any theorems, but if you want to run with that and figure things out, cool. Anyway, okay, so this is without even using those inclusions. This is just, right, this is something that you can compute using your existing toolbox on any weighted graph. All right, further, I can use filtrations to get rid of that stupid nonlinearity that we ran into in the beginning. Okay. So monotonic transformations destroy everything except maybe ordering. If I don't know what the monotonic transformation is, I can't trust the values, but I can trust that if A is less than B, then f of A is less than f of B. That's the definition of monotonicity, right? So I'm going to retain only the ordering of elements. I'm going to collapse all the information in the matrix except what order they came to me in. So I'm always going to think of matrices as being the following. The first entry is 1, the second entry is 2, right? And by first and second, I mean in order of increasing value. Somewhere in the matrix there's a 1, somewhere else in the matrix there's a 2, somewhere else there's a 3, yada, 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 up through n choose 2. Right? Every matrix, that's the only thing I can see. Okay. So if you're a mathematician, you might like to think about this as the equivalence class under the orbit, but if you're not a mathematician, just don't read this. 1, 2, 3, 4. Okay, so I just take decreasing edge weight and I replace it with, I like to use edge density because I like my numbers to run from 0 to 1 so that I can compare different sizes of graphs. You don't have to. You can just put this as 1, 2, 3, 4, 5. I'm going to evenly space these things. Okay, right? So just to be clear, now that I've done that, right, I've forgotten everything except the ordering, so any damage you did to me with your special nonlinearity, your nonlinear response to my input, is gone. So hooray. So I no longer have to worry about this. This is like the invariant under change of basis. This is invariant under change of values. Wild change of values. Okay. Filtrations, straightforward. Simplicial complexes, also straightforward. It's like a graph. It's like a network, except I'm allowed to put in bigger things. But it's not a hypergraph, because there's a requirement. And the requirement is this. If I put in that tetrahedron there, I'm required to put in all the triangles and all the edges and all the vertices that are its faces. That's all. Okay? So downward closure. Once I've included this triangle, I have to include these 3 edges and the 3 vertices. So I can specify one of these things to you just like a graph. I could give you a graph by just giving you a list of edges if I wanted to, assuming that there aren't disconnected vertices. Here, I can give you a simplicial complex just by giving you a list of maximal faces of all various dimensions. No big deal. So they're a natural data structure to use even if we know the underlying thing is a network. Because activity, coactivity, for example, in a network, right, a bunch of neurons firing together, that's not a pairwise thing. That's, I observed these 7 neurons all doing the same thing. Right? So I shouldn't say it's this pair and 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 this pair, which is what I have to do with a network. I should say it's this 7 objects. So this is really the natural language. And if these 7 objects were all co-firing, any 5 of them were, so all the faces should be included. Right? There's no extra data going in here. And this is just to illustrate that, the power of that, right? So if I observe these 2 neurons co-firing, the blue and the red, these 2 blues, and then the green, the blue, and the red, the resulting simplicial complex would look a little bit like this, right? I've got these 2 co-firing, these 2 co-firing, these 3 co-firing, and I'm differentiating the fact that these 3 co-fired and the fact that I didn't see those 3 co-fire. Okay? So that's the kind of data that we can extract. So even, here's the best part, if you give me a network, I can still build you a simplicial complex, and I can do it without making any choices, which is what I like to do because I don't know how to make choices. What do? I look for the cliques. Right, we've heard cliques a few times already. They turn out to just be fantastic. So if this is my graph G, the clique complex is literally the simplicial complex where I put every clique I possibly can in as a simplex. Right? So I made no choices. I chose them all. Okay? So, yeah, it's the canonical simplicial complex you recover from a graph. And I like that word because it means that I don't have to do anything. And so the data of this clique complex is the data of the graph. So way back to the beginning, if you give me a correlation matrix and I'm looking at a threshold, I already have a clique complex. I don't have to do anything new. Okay. So simplicial complexes, easy. This is also easy. All right. Homology. It's actually, we have to unpack it a little. So homology is how we measure simplicial complexes. What have I got? I've got a clique complex, so I'm going to think about this clique complex. I'm only going to think about some of the vertices because they get kind of big. So, all right, what if I were just looking at this thing, I would want to see that there's a hole, and then there's this two-dimensional hole surrounded by all those triangles up there. Okay? And I want to be able to count that information. I want to do it in an automated way on a computer because, frankly, when I've got 100 vertices or 1,000, I don't want to get involved. So what I'm going to do is I'm going to stratify the clique complex by listing the cliques of each dimension. Okay? So I've got zero-dimensional cliques, which are just vertices, right? And 1, 2, 3, 4, 5, yada, 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 yada. I've got one-dimensional cliques. Those are the edges, right? I've got two-dimensional cliques. Those are the filled-in triangles. I have no three-dimensional cliques. There are no tetrahedra up there. There are no 15-dimensional cliques anywhere in there, so it's zeros all the way up. Okay? So I've just got this stratified collection of sets indexed by dimension. So far, so good? Okay. Now, I told you that there's this boundary, this notion of a boundary. I'm going to need that where I'm going. And there is the notion of a boundary, right? Clique 1, 2, 3, that's that triangle. What's its boundary, naturally? Well, it's that edge 1, 2, that edge 1, 3, and that edge 2, 3. Ah, okay. So the boundary of 1, 2, 3 is three things in this. Oh, I'm not going to be able to make a function out of this because it would have to be a multifunction. Nobody likes multifunctions, right? That's way too complicated. So these dotted lines are never going to work out. I need to go somewhere where I'm allowed to make this a function. So I'm going to follow one of the guiding principles in my life. It says that vector spaces are better than numbers. So I'm going to move to the world of vector spaces. And for simplicity and also for computational purposes, I'm going to work over the field of two elements, which we heard about in I think Matt's talk this morning. Right? My only numbers are 0, 1, and 1 plus 1 is 0. It's the best place to do arithmetic. Okay, so I had the empty setup here. That's going to give me the 0 vector space because there's nothing for me to build a vector space on. Here, I had all these triangle cliques, so I'm just going to take a vector space with basis corresponding to the triangle cliques because it's the only thing I know how to do is just, right? If you give me a set, I can give you a vector space with that set as a basis. Here, right? So what's a vector, right, so okay, so just for the people in the audience who might not be totally comfortable with abstract vector spaces, right, I'm just specifying to you a list of things that you're allowed to add together using the plus symbol, right, there's nothing like, and this one plus this one is literally, that's what it means. It means E12 plus E23 is E12 plus E23. There's no rule. I'm just allowed to do that, which is how I'm going to get around this problem back here. Okay, so I'm just going to take the vector space with those elements as bases, and then these are called chain groups, and then I'm going to press the right button over and over, and nothing's going to happen because my computer is locked up. Close the program. Apparently, Acrobat Reader decided to quit. Decided it didn't want you to learn about this stuff because it's dangerous. So we'll try again. Okay. Oh, that's great. It's cut off at the bottom. All right. Hopefully that won't be a problem later. Sorry, technical difficulties. Hardest part about this entire thing is getting the computer to work. That's going to be terrible. Okay. Sorry. Okay. Now I can define these maps, right? Where does E1, 2, 3 go? The boundary of E1, 2, 3, well, it should be 1, 2 plus 1, 3 plus 2, 3, right? So it's exactly the sum of the things you want it to be a sum of. That's perfectly okay. In a vector space, I'm allowed to add things. So now it's not a multifunction, standard function. In fact, linear transformation, great. That's what I like between vector spaces. So let's try it out. So let's see. I'm going to compute, right? I'm going to take some edges, and I'm going to compute their boundaries. So 2, 3, that has vertex 2 and vertex 3 as its boundary, right, geometrically. And I can add also 3, 4, and 4, 5, and 2, 5. and compute their boundaries. And since this is a linear transformation, I can just add those all up and see that the boundary of that sum of things is 0. But notice that's exactly a closed cycle up there in my complex. Okay, sensible? I'm not sure if you're either like, yes, this is so dumb I can't stand it, or where's this guy coming from? He's from some other planet. Yes.
Do you have orientation on the edges so that there's some minuses?
No, because I'm working over Z2 to prevent you from ever having to worry about signs. That's where the simplicity is. Yes. This is standard homology. Yeah. Yep, it's just standard homology of spatial complex. Turns out to just be adding things up formally. There's nothing really complicated about it. Okay, so I'm going to declare everything whose boundary is 0 to be called a cycle. And of course, mathematicians have to have a fancy word for things, so I'm going to call the collection of all things with boundary 0 the kernel, which, right? Call it the null space if we were in linear algebra, but technically you don't have to have vector spaces, so whatever. Right, so these things are called the cycles if your boundary is 0. So here's an example. 1, 2 plus 2, 3 plus 1, 3, right? You can do that in your head. Those things are all going to cancel because each vertex appears twice. So 1, 2 plus 1, 3 plus 2, that is not a hole. Okay, so I made a bad definition. I need to fix that. So why is it not a hole? Because somebody came along and glued a simplex into it. So so that is the boundary of another simplex, right? So I'm just going to say, well, those things are not simplicity, or those things are not holes. So the image of the boundary map coming from above is called is a subspace of the kernel. So there would be a little activity alert down here if you could see the bottom of the screen. That's the activity. And those are the boundaries of things of one dimension higher. And I'm going to say, tell you what, if you're the boundary of something one dimension higher, you're not really a cycle. So I'm going to take all the things with dimension 0, and I'm just going to kill off anything that happened already to come from above. Yes? They are linear.
It's not clear when I look at this that you have, like when you had the E2 and E3 together, you have an image for that.
Mm-hmm.
It's not clear that there's an image for E2. There's an image, you know,
For E2? Yeah, so there's the 0 vector space sits down here.
yeah.
So the boundary,
Every basis element at each level
has an image.
Okay.
Yeah, so right, we have no negative one-dimensional simplices, so I just have to hit 0. There's no choice. Yeah, thank you. Sorry, I'm going to take 30 seconds and fix this because that would have been, I think, clearer if you could see the 0 vector space sitting down there. Any questions while I'm fixing this machine? Oh, where'd it go? It is, in fact, Windows 10. My Linux laptop is too heavy to carry.
Yeah, I like it that when you go to the field of two elements that you don't have to worry about orientation. I wonder if that would be nice for teaching it or not.
It is fantastic for teaching it. It is also fantastic for computing things. There's really no downside to the field of two elements except that the Mobius strip is the Klein bottle is an orientable object. Oh, you're kidding me. Oh, okay, I see. No, don't. Sorry. It wants to update my Acrobat reader. That's what's going on here. Okay. Haha. Yeah, see 0 vector space down there at the bottom. Okay. So, that's homology. I'm allowed to add up. Right now I'm allowed to add simplices together. No big deal. Or cliques, if you don't even want to think about simplices, you can always think about cliques in a graph. Those are the objects I'm starting with. I'm allowed to add them up, and I can look at the boundaries. And if the boundary of a collection is 0, then that thing is a cycle. And if it didn't come from something above, then it's a cycle that's actually a whole. Okay. Cool. So it's convenient to summarize that information, right? So that's a sequence of vector spaces. These things are vector spaces. It is not necessarily the case that everybody cares to think about vector spaces, although in the activities wrote down, I really think that that's worth unpacking for understanding. So instead, you think about the ranks of those vector spaces, just the total number of cycles, right? So here, I've got Betty 0 is the number of components, right? That's how many vertices I have that don't differ by an edge. So that's just the number of components, right? There are 2 1 cycles, and there's a big 2 cycle here, which bounds the 3-dimensional volume, right? Yep. So it's Betty 0, Betty 1, Betty 2. So Betty 0 is the number of components, which I didn't draw here because it's pretty clear. Betty 1, there are 2 distinct holes in this thing.
Okay, that's the identical, that's an identical.
Yeah, yeah. Yeah, sorry, same thing. I needed to highlight 2 different things here. And then there's one 2 cycle sitting in there. Okay. So this is the statistic I'm going to use for simplicial complexes. Why? Because these are those things, right, back at the beginning with that rectangle. Those are the things I want to see whether those things are around or not. Okay. Whew. So, there. You are all now official topologists. You know our basic tool about as well as most people who take the first year of topology do. So, and actually probably better than many people who actively use it. So, I know. I meant me. I have to use this to do work, by the way. I just flip through these slides when I have questions. Okay. Combining all 3 of these things, we get what's called persistent homology. So here's the idea. I have a filtration on a simplicial complex. So now I'm allowed to put numbers on anything in the simplicial complex. A filtration on a simplicial complex requires that if I add a face, I add all of its boundaries. So there's some rules, but we're not going to care about that too much. Okay. So I've got a filtered simplicial complex. At each stage, I can compute the homology, right? So I'll get some H0, some components, some H1, which are loops, some H2, which are volumes. There might be more stuff going on. We'll see some more stuff later. As I go, these things, right? So these components are getting connected up, so things are dying off. Right? Eventually, I get to this point out here, and I've only got one component left. Okay? And I could use those inclusion maps from the filtration to tell you which things are dying, right? As I go along, right? If it was a component here, that's a homology class, and I can follow it through a map on homology that comes from the inclusion map on this complexes to tell you that this one on these 2 pieces became this one here. So these 2 things collapsed as I went from this stage to this stage. So one of these homology classes died on the way over. Yes?
How are you picking the order of the edges again?
These things down here? Oh, up here?
Filtration, you said
There's just a weight. Yeah, there's some
it
weight.
seems like it would be really sensitive to like changes in.
That's a fantastic question. So in fact, it is not. If you epsilon perturb, so this is the theorem that got all of this started. The stability theorem of a guy named Cohen-Steiner says that if I take this object and I perturb the weights by epsilon, I perturb in an appropriate metric space structure on this, I perturb the answer by epsilon.
Is that with probability like?
No, just true. Like it's a contractive map.
So you can't even construct a weird example where it fails.
Nope.
Who's that?
Cohen-Steiner. Cohen-Steiner. There are more modern versions, and I'd be happy to give you references at the end that give better expositions than the one in that paper. Yes?
I'm just confused about what your bottom graph represents, what the changes along the horizontal axis are
Right.
versus what the
So-
shifts within like H0.
Yeah. Okay. So this is my parameter, right? My filtration parameter, right? So this is the number of edges that I've added, or the number of simplices I've added, right? And so then each of these is sitting at such a place. At each place along this line, I'm computing the homology, right? And so here I might have H0, or right at the beginning I had H0 with rank 12 or something, right? At this point I have rank 1, 2, 3, 4, 5, 6, 7, 8, 9, right? And so certain components are going to persist a lot longer than other components. Right down here I have cycles forming, right? So this square forms. By the time I've gotten over here, this square is dead because it's been filled in with a simplex. right?
Make sure I understand this. Take that second blue line from the left.
That one there. That one.
Yeah, that one, the dotted one. Then at that snapshot,
Yep.
there are 6 connected components in
Yep.
your simplicial complex, and there are
And two loops.
2 cycles.
Yep.
And then at that little place up there, the 5th one, at some point you have a 2-dimensional hole around.
Yeah, it's right around there, I think.
And you're adding edges, and then you're adding.
I'm adding this. So in this case, I'm just adding simple C's in sort of a way designed to make this picture look good. In a network setting, I'd only add edges and then fill in the cliques because I have to, right? The clique complex tells me the rules, right? So, but the point is I can compute homology of any given simplicial complex. By giving you a filtration, what have I given you? I've given you a sequence of simplicial complexes. I'm going to compute homology.
Bar represents an n-dimensional hole.
That's right.
So how long
And the length of the bar tells me how long it lasts. So we call the left-hand endpoint the birth and the right-hand endpoint the death of a cycle. Yes?
Well, in the second picture there, it looks like there are 3 cycles.
There's one there and one there. And that one is yellow. There's a yellow simplex sitting in there. Sorry. It's a little bit pale, but yeah, that's filled in. Sorry. Yeah, it's a triangle that dropped. All right. So we don't need so it is physically impossible to give you a very deep understanding of this process in an hour-long talk if I want to talk about anything other than persistent homology. So I put that all in the, I put a fairly decent example, I think, in the activities, which I would love to help you all think about if you want to think about it. It's these, like I said, these ideas are simple, and you just kind of have to work through them. And also, please don't read the introductory papers. They are aimed at mathematicians. There are very few. There are a couple nice books that have recently come out, and but most expositions are still aimed at people with graduate mathematics. That's just. There are that's in process. It's a young discipline, and also we still don't know how it works. So people are kind of hesitant to write an exposition. 5 years from now, people will be like, "Why did you put this in your book?" Okay, so now here's the cool bit. If you give me a probability distribution on symmetric matrices, and I do this thing I just told you about, right? I take the matrix, I consider it to be a network, I consider that to be a filtered network, I consider each of those to be a clique complex, I compute persistent homology, that gives me one of these barcode things I just drew you. Okay? If I give you a probability distribution on symmetric matrices, I just shove that entire thing into this machine. What comes out? A probability distribution on this thing. Okay?
Just to make sure
Yep.
you don't care about the order of those components.
No, that's just completely arbitrary. In fact, yeah, what these things represent is actually very hard to describe. That's also in the activities. All right. This is interesting because if I have observed a system and I want to falsify whether that system is of a particular kind, what do I need to do? I need to have a null hypothesis, right? I need to have something I can check. I need probability. So, by the way, that theory is still in process. Nobody has really settled that theory in. So I want to say that's active research, and if you are interested in this, we need so many people, especially people who can think about that, because it's what we need. But we don't understand, in fact, probabilities on persistence diagrams and barcodes. For example, the mean is not well defined. So instead, we're going to look at some summary statistics. And the simplest summary statistic is that vector that I told you about, right? The Betty number vector. Instead of looking at all the bars, I'm just going to look at how many there were. So now what have I got? I've got a vector-valued function on the filtration. So this guy, Matt Kale, was the first person to have this clever idea. He didn't really realize he had had it at the time. His paper is about the structure of these things, and this is just a shadow, but it's a tremendously powerful idea. So he took, this is a picture from his first paper, 100 by 100 matrix, and he took the entries to be IID uniform. Okay? And so what happens? So Betty 0 falls off almost immediately as you start adding edges, right? The system closes up. 2002, maybe 3. This is very, very new stuff. Right, this is the very first paper that people were thinking about this stuff. I saw him give a talk on it when I was in graduate school. It was a while ago now, but not that long ago. Betty 1, right? So this is fascinating, right? So then the 1 cycles start to show up, and then they die, and then the 2 cycles, and then the 3 cycles, and then the 4 cycles in order, right? This is, what is this? This is a matrix, and at every threshold, I've just taken entries to be IID and said if you're above this, right, above this number, then you appear. Oh, so that's a bunch of Erdős-Rényi random graphs. Right, this is a sequence of Erdős-Rényi random graphs of increasing probability of having edges. And at any given edge probability, the homology is really, really predictable. That's fantastic. Right? I think this picture is really secretly responsible for most of people's interest in using this for data. Because it's too good. And they grow exponentially. Like if you had, as n goes to infinity, these things shrink by about half as you go farther right, and they grow. This was as far as he could compute this at the time. This was using custom software. We can do a bit better now. However, he also used not the values, but the edge density down here. And remember, any two probability distributions are related by what? A monotone increasing function, inverse of the CDF composed with the CDF, right? This is like fact from probability theory. So in fact, he didn't have to choose uniform. He could have chosen any probability distribution on the matrices. He got the same answer. So back at the beginning, that thing when I was picking on Wigner's semicircle law, right, for n equals 100, I get always the same answer no matter what terrible thing you've shoved me through here. So I can always recognize IID matrices. They look like this. Second example, this is my my example that I want to look at, right? This is the activity I'm looking at geometric, right? Are the neurons points in space? Is their correlation dependent on distance? So I'm going to take points, say in the unit cube, in some dimension. I'm going to take correlation to be the negative distance. Why? Because I just need some decreasing function of distance. And it turns out I don't care which one, because monotonic increasing function. And I'm going to take n equals 88 because the data I have laying around for this particular example has 88 cells. So take a bunch of these, right? These matrices, shove them into the machine. Different picture. Okay. So this gold is Betty 1, red is Betty 2, blue is Betty 3, and then these numbers here are the dimension of the cube. So in 5 dimensions, I get these pale ones down here. In 88 dimensions, I get those ones right there. Okay, and the thing that I want to point out is that unlike the ones that were increasing exponentially before, these are not. They're in fact falling off exponentially. Very nice. And also, in a second, we'll see there's a tremendous difference in their scale. So this tool actually does see the difference between these two situations. And the reason is back at the beginning with those 4 vertices, right? If I form a cycle here, it comes with a death sentence. Right? At some point, another edge is going to come through there and fill it all in with cliques. Right? In the random case, I form a cycle and I get to sit around for half an hour and it's going to be a cycle. Right? So they live forever. Some of them do. So that's a great, right? Very, very different things. Whew. Okay, so remind you of the picture. This is what we want. I'm going to check not in brain data first because that's really messy and hard. So what if I just model place cells with inhomogeneous Poisson neurons? Right, so I take the actual place cells from the data, those heat maps I computed, and use the mouse's actual trajectory, and then construct inhomogeneous Poisson neurons so that I can tell that, so then only the geometry of the situation is what the tool should be picking up. I've gotten rid of all the neural confounds. And lo and behold, that was a very complicated ringtone. Lo and behold, look, they're sitting right on top of those. Cool. They're sitting right on top of the d equals 5. It's supposed to be 2-dimensional. Here's the problem. A lot of place cells are garbage. So these are some of the receptive fields. Yep. Right, that bottom one is d equals 5 in my picture. Yep. Simulating neural firing as an inhomogeneous Poisson process based on the heat map.
So those lines are neurons firing?
Yep.
But they're also coded as to which one is which.
Yeah, so different neurons have different pictures. Right, I had 88 cells and each one has one of these.
So in that picture with the time, can you tell
Yep.
that?
That's just for one.
Yeah, this is just this one neuron that I've got a picture of here.
Oh, I thought there were 2 neurons.
No, so this is just, this one neuron has this little blob up here. Right, so that's the problem. This is real neural data. Even my simulated real neural data is not two-dimensional because the geometry was a lie. Because there are these terrible cells that don't have the geometric structure. I thought that, right, so my model was wrong because these things are, but it's pretty darn close. I couldn't get the dimension. Yep.
What's that picture of again?
This here? Yeah, okay. So we took the original rat, we have the original rat experiment, which is the spiking for each neuron along with its location in time. So let the rat wander in the environment, right? And as it went, every time, so I cut the environment into 1,000 by 1,000 grid, actually. And so in each box, I just recorded if there was a spike in that box. And then I normalized that number by the amount of time the rat spent in the box compared to the total amount of time. Then I convolved the entire thing with a Gaussian. Right, that gives me, so that gets rid of my edge effects on those little boxes. So the result is a rough estimate of how active the cell is at each location. Right. So then I constructed synthetic data, which doesn't have any of the neural confounds that would be gotten from like recording with probes and things in order to get, right, just geometry. But it's just geometry built out of actual recordings. Any other questions?
Do we know there aren't more specialized place cells somewhere else in the brain?
Nope. In fact, there are lots of places where we record location. This just happens to be the one that people have studied really well, so it was a nice proof of concept choice. I can tell you a little bit about another place in just a second. But I do want to show you that, so I didn't get D equals, oh, question? No. I didn't get D equals 2 because place cells don't actually all work, I didn't do any pre-processing, I didn't throw away bad place fields. So, right, because in the back of my head, I'm pretending I don't know any of this is here. So I'm not allowed to do that. Okay, and then just to check, maybe you say to me, Chad, maybe there was something else besides the geometry. So what happens if you destroy that geometry? I did the same experiment, but I shuffled these place fields more finely than this. This is just schematic, right? So you get and then it's not, in fact, this is the weird thing. Betty 1, this is Betty 1 here, is actually in the geometric regime, even though I destroyed the geometry. So it turns out that I need 2 cycles and 3 cycles to really prove to me that the thing I'm looking at is geometric, which is the first time I'm aware of that anybody's used Betty 2 and Betty 3 explicitly to determine something about a system. So, but you need it. You can't not have that data. This is not a sensitive enough detector, but these ones are. So 2 cycles, right, spheres, and 3 cycles, more complicated spheres. Okay, so now I'm going to do it on the real data, because if I didn't do that, then you'd all say, go home. So I've just accumulated things. So here's the random networks. I wanted to show you how consistent these are. So these Betty 1, Betty 2, Betty 3, that's those little envelopes there are 99.95% confidence intervals. I did not include the other 50 trials because I was told that would be cheeky, but they also fall in there. So as far as I've just the probability of ending up looking like that is 1. Geometric, these are means because it gets really ugly if you try to put the variances. The variances are big enough that it's hard to detect dimension even in the case of pure geometry, but these have order 200 for Betty 1, this has order 60. These have order 1,000 for Betty 3, this has order 20. So you can home apart. Here's what happens when you take actual rat place cell recordings, the ones that I built the original place fields out of, stick them in, lo and behold, sure look like that. They're a little bit chunkier, Betty 3 is, but they're pretty darn close. Okay, and then in order to make this even simpler to compare, right, because the scales are so dramatically different, we just integrate the curves, right? Get a number instead of a curve to compare. So I'm going to just integrate each of these curves, and so G is the geometric regime, and S is the shuffled or random regime, and you can see data falls right in there. You say, okay, Chad, great. You went to a ton of work to show us one thing that John O'Keefe told us 40 years ago. Great, thanks. Okay, place cells have place cell structure. Here's something John O'Keefe did not show you. So In spatial navigation, where they were studying it, lo and behold, you do this experiment, everything falls in this geometric regime. Wheel running. So now the rat's running in a wheel, so we've gotten rid of spatial cues. There's no visual cues that the rat has a location. Now it turns out that it's known that hippocampus produces what are called episode fields while the rat's running in a wheel, but these are linear, okay? So it's not quite the same structure. But we do pick up a geometry there, which one would expect from the episode fields. And then there's this. When the rat is asleep, its hippocampus is still producing this geometric structure. Okay? No stimulus, no location information. Right? That you can't tell without this tool ever. Why? Because you have no behavior to correlate with. Right? So this is new. So hooray, topology did something for neuroscience. So what's the conjecture? Conjecture is that the geometry is a fundamental property of the way this network is wired. Its activity is always going to look this way. And I'm happy to say that we made this conjecture about 2 years ago. This year in February, I went to COSINE, Computational Systems Neuroscience, best neuroscience conference for quantitative people. I highly recommend it if you like this stuff. There was a lab there that essentially did an experiment that corroborates this conjecture by showing that there are tone fields in the hippocampus, which is clearly not something that actually evolved. Right? The rat was given a varying tone that it could control, but it was stochastic, so it wasn't like there was time or any other information. The rat had to navigate a quote-unquote tonal maze to get a reward, and as it did so, fields appeared at various places in the tonal maze. Right? So what's the hippocampus doing? It's taking aggregate information from the brain, and it is arranging it in a geometric fashion for access by other places. It's giving information coordinates. So maybe it's doing MDS, I don't know. But whatever it is doing in there, right, it's doing it all the time. So that's fantastic, right? And maybe that suggests to you that place cells are kind of a lie. They're the first thing that we correlated the behavior against, or correlated the observations against, but maybe they're not the actual thing that that area is doing. Although it could be one of the outputs that we use a lot, right? Could be we got really good at figuring out locations, so that's what the hippocampus started doing, and then we started overloading that circuit as we needed more and more and more complicated inputs. Right? A lot of theory that needs to be done to understand that. Whew. Okay, so I am out of time. So we made it to the punchline of the big thing. So I would like to pause and take questions and stuff. Yeah.
Have you, well, so in this system, it's the spiking relative to the phase of the data.
So in
Have you ever tried to include that in your analysis?
So these are all situations where the rat has theta. So if you remove theta, in situations where that doesn't have theta, you get a degraded geometric signal. Also, I should say, on different time scales, different behaviors give you different amounts of geometry. So this is a 1 second cross correlation window, which is enough for 8 theta cycles, so we want to aggregate across those. If you shrink down to about theta, both wheel running and REM sleep start to lose their geometric structure, but spatial navigation retains it. Which
Have you ever tried to actually keep track of the timing of the spikes with respect to data? Just like in the
yeah, the precession and all that.
sleep, the precession and all that kind of stuff is one of the important parts.
Yeah, absolutely. So the ripples, which are the part that are driven by are about 8% of the depending on who you ask, 8 to 12% of the total spiking during REM, which shouldn't be enough to drive the correlation structure. So that was one thing that we did check, is that if you sort of right, that's not what you're seeing there. I mean, it might influence what you're seeing there, but I think it's actually the other direction. I think playback is a result of the structure, not the other way around. Like the whole emergent behavior of playback, even if it serves a functional process, is sort of an epiphenomenon of the fact that the system's just wired up to have this kind of structure in its behavior. That's personal opinion. I can't prove it.
It's interesting that it's an ancient mnemonic technique is to have a spatial structure like a tree or a palace or something. So anything that you want to memorize, you put it at different locations in this imaginary structure in your head. So
No, I think that's actually, I love that stuff. I'm not any good at it. I've never been able to make it work. But people being able to do it is great, this mind palace stuff. And I think it has to do with the fact that associative memory and spatial location are the same circuit, right? That has to be the reason. So, which, again, goes back to the thing I was saying before, which is that I think that probably we learned to navigate and then overloaded that with other things, including long-term memory formation. So, but again, personal opinions, I can prove none of these things. So, yeah. Yep. Something like that.
So you said that some of these are these simulated?
Nope. These are constructed from data.
Okay. So you said that some of them are false.
Well, they're just not geometric, right? So there's no, they're not a Gaussian centered at a point, which was what I went in claiming that place cells were, right? They're a mess, right? This is what I want them to be. This is what I got.
Go back one more time.
Yep.
What's that white?
Oh, that's a cartoon animal trace, right? So that as the animal moves, this inhomogeneous Poisson process down here is generating based on how hot the heat map is. Sure. Anything else? Okay. Yeah. I'll tell you a couple more things. So, okay, you say, Chad, that's great. You've got a tool that does exactly one thing. If we happen to be looking for another hippocampus, we know where to go. So this is my student Ann Sizemore, who was fantastic. She's now at the Broad doing cancer research while she prepares to go to graduate school next year. By the way, if you want a fantastic student, lure her away from her job. Yeah. Well, I'll train her. She was a master's student, and she's got 3 papers. So she's the Broad Institute at Harvard and MIT. It's a cancer.
We can lure away from there.
Well, I mean, she's only got a master's degree. She needs to go to grad school somewhere that'll teach her to do math. So, okay. So she took this group of model networks. These are just standard things from the neuroscience literature, and then and then some geometric networks in R3. We wanted to compare them to. These are standard things people use as null models or ideas of how network function is supposed to be constructed in the neuroscience literature, right? But I want this. I'm thinking of this as a model for any other network that you might want to run along with. Okay. So she took this 14 network list, shoved it through the machine, and we discovered there's a roughly 2-dimensional structure. You can take this entire set of topological features, and we computed more than just the Betty numbers here. We did a fairly in-depth analysis. But you get a picture of the space of all of these networks, and they sort of smear along an arc like this. From disordered networks, things like Erdős-Rényi networks, maximum entropy networks under various conditions, down here to things that we call coned networks, which are, they have like a hub that is connected to absolutely everything. Okay. I don't believe in these 4 classes of networks. These are just the 4 classes of networks that fell to the biology networks. What I believe in is that there's this picture of the space of all networks. Up here you have things that have a ton of cycles and not very many cliques. Out here, right, they've got very small cliques that are just all over the place. Out here you have things that have one massive clique, one thing that just talks to everybody everywhere. And in between you've got various sort of classes of networks. Some of these things, some classes of networks, some of these, this is probability distribution thing is back, right? There's sort of smears where different network models live in this space, and they live along this big kind of arc in here. So this is just, schematic picture, but this is the result. You take all those networks in, and so that tells you what this tool is seeing, right? It's seeing the difference between these kinds of network structures, which is not every kind of difference between network structures, clearly. It's just the one kind of picture. And I don't want to go into the tremendous details, but here's how I think you could use this. Say I have, so here, our 4 data networks, and here are the 4 classes that we extracted. And so we just computed in this network space the distance between each of the 4 data networks that we're interested in and each of the 4 network the centroid of those 4 network classes, right? And what do we see is that class 4 is, let's see, class 4 is far away. Oh no. Yeah, we're very close to class 2 with all of our data networks. So then we could look inside the class of data networks, look at the centroid, the mean of, right, look at the Frechette mean of where that each of the probability distributions for each of these classes of networks is located, and compute the distance to each of these, right? If you're far away, you're probably not a good model for the data matrix, right? This back to topology is good at invariance. It's good at telling you what something isn't. So in this space, if you're far away from, if your network of interest is far away from the centroid of a model network, then you're probably not that network. So this tells us that these things are best modeled by things in class 2, probably. So these are Kuromodel oscillators. I think that's a some sort of protein network, and DSI is a brain network. Right, and then these all have varying levels of nearness to each other and varying levels of nearness to our model networks. This is a way to do model validation on network types. Right. So you can take your favorite model networks and plug them in here and check, which as far as I know is something that people don't really have solid methods for doing. So anyway, this is going to appear in the Journal of Complex Networks in the next next edition, and then Anne just kept going because she never stops. So I couldn't do the structural network I wanted to do at the beginning, but this has got an 83 nodes. It's a whole brain network. And so we computed the persistent homology here. This is a different way. We're plotting birth, comma, death here, right? So you took those bars, and there are 2 coordinates that determine the birth and the death. So let's plot those. This is just if you live on the West Coast, you use the bars. If you live on the East Coast, you use these, and there's a feud. Applied topologists shooting each other. It's terrible. So birth and death. Okay. So there are some 1 cycles and 2 cycles that show up in this network. And you can actually go back and look and see who they were. This is one of the other things this topology stuff gives you. It gives you the ability to go back and say, hey, what in the original network produced this cycle? However, giant exclamation point warning, homology classes are equivalence classes. I killed off everything that was a boundary. So I get non-uniqueness of representatives. It is almost always the case that it is impossible to correctly decide what the representative should be. But here, we were able to do it, and I'll tell you why. Because these things appeared early, and they didn't have any boundaries attached to them, except these ones. So these 2 on the left didn't anyway. So this is a cortical-subcortical cross brain network. That's the kind of thing you might expect to be a powerful subnetwork. I don't want to get into the neuroscience end because honestly, my cognitive neuroscience is not great. I'm still working on it. This is really cool. This is like the what-where pathway in vision. And this is a 2 cycle, which I'm super excited about. But we did get a small amount of choice here. So these 2 vertices were strongly connected, so they created an equivalence class here. But that's okay, because these 2 things are homologous, or share similar function in the brain. Similarly, what's that? Oh, sorry. By the way, so topology and neuroscience and biology in general, I feel like there was the paths diverged in about 1910, and I feel like when the topologists left, they took the dictionary from the biologists, and they're like, we're going to use these words. Because it's just nonstop. Cells, complexes, nerves, germs, sheaves. It's just, it's almost malicious how biology is not malicious, topology is malicious. So, but anyway, so these are strongly connected groups of 5 to 10 brain regions each, right? These are actually almost cliques, but they're good cliques. They're cliques of things that serve the same purpose. They have strong connections on purpose, and they still form this nice sort of control cycle. So you show these to cognitive neuroscientists, and they go, oh, those are the kinds of circuits that we would expect to find in the brain. Great.
Is that the resting brain? Usually in functional image, so usually there's a two condition.
So this is DSI.
So what?
This is DSI. It's structure. White matter tractography. So, so we're working, DSI, great question. So anyway, I wanted to say the cycles that you find are robust. So there are 3 scans of 8 individuals, and you see all of these things over and over and over. Right? These aren't like an artifact of the one bad scan. And if you do some other things, so fMRI. Greg Henselman, who is a student of Rob's, will be starting as a postdoc at Princeton in the fall. He's fantastic. So Greg is responsible for the fact that if you want to use this tool, you can. Because when I was doing work with this stuff a few years ago on the stuff in the first half, it took 8 hours to do these 90-node networks. And we couldn't get the full answer. We had to kind of cut it off. Greg just put a tool on the web. There's a link to it in the activities. In the Julia language, if you don't use Julia, you should. It's fantastic. It's like MATLAB, but free, and it's as fast as C, and it's magic. Greg wrote a software package in Julia called Irene. Here we have 264 region power parcellation, so it's just some parcellation of the brain. That runs in about 10 minutes on my laptop. So, different world now. But this is now computable. Persistent homology in these by the way, these are the hard persistent homology computations. If you do point clouds and things that people do otherwise, you can rely on the geometry to cut down the computation. These brain computations, you've got no sort of in like that. So you have to just kind of power through, but Greg is very smart. So he made this computation possible. This is, as far as I know, the biggest persistent homology computation ever done. We took 780 of these 264 region, 264 by 264 matrices, and computed their Betty numbers. And this is cool. Resting state fMRI, what does it look like? Looks geometric. Don't know why. So this is a sneak preview. We have no idea what this means, but looks cool. Yeah. Because that takes longer. So these things, right? It's polynomial with each one of, right? So Betty 1 requires us to compute 2 cliques, cliques on 3 vertices. Betty 2 requires cliques on 3 vertices. So computation now. We could do Betty 4. We probably could not do Betty 8, at least on something as big as this. If you want to do a small graph or local computations or whatever, you can. So, and then, yeah, we get variation across subjects. It's fantastic. So anyway, thank you all for listening. This is a survey paper that Rob and Danny and I put together. It's in the Journal of Computational Neuroscience. It's aimed at neuroscientists. But if you're willing to read around the neuroscience examples, because you don't care about neuroscience, it should be readable by, hopefully, pretty much anybody who's interested. Not a lot of hard details, but pointers to where you can find that information. So, anyway, thank you.